Hi, my name is Jesse Butler, and this is my photosynthesis video. I'm going to start off with the light reaction cycle. And to start off with, on the light reaction cycle, I'm going to start with the thylakoid space, which is a space inside the thylakoid. And the thylakoid membrane is an internal system of interconnected membranes that carry out the light reactions of photosynthesis. And then we have the stroma. The stroma is the supportive tissue of the epithelial organ, tumor, gonads, etc., consisting of connective tissues and blood vessels. Here you see the photon of light is entering, and there it enters and it goes to the chlorophyll. But before we get there, we have 2H2O, which upon the process of the oxygen evolving complex, you are taking away two hydrogens and you're going to release the O2. You're going to release the O2. And all of that in the oxygen evolving complex, including the chlorophyll, is going to bring you to an electron. This is the portion which is considered photosystem 2. From the electron, you got plastoquinine. I know I butchered that, but from there is a process of a hydrogen entering and leaving, going to a higher populated hydrogen area. The plastoquinone is then taken to a plastocyanine. From there, you're going to add another photon of light. This area is considered photosystem 2. From there, you're going to go to ferredoxin, and ferredoxin is going to go to a process of ferredoxin NADP reductase, which is the last enzyme in the transfer electrons. From there, you will see the NADP plus evolve into NADPH. It's basically going to add a hydrogen. Next, you're going to go, this is the end of the light reaction cycle, where you get to the creation of ATP. This is where the ATP synthase takes place. ADP is going to be involved with the hydrogen, and it's going to form, ADP and the phosphate are going to be involved with the hydrogen, and they're going to form ATP, and that's ATP synthase. This is basically the light reaction cycle. This is the Calvin cycle. As you can see, it starts off with 3 CO2s and then 1 carbon added to it. It begins in the cycle and it goes to 6 phosphoglycerates, and the 1 carbon becomes 3 carbons, and then as the process continues, you're going to add 6 ATPs, adding 6 energy, and you're going to release 6 ADPs, and you're going to get 6 1,3 biphosphoglycerates, and still going to be a 3 carbon. And after that, you're going to see the addition of NADPH, which is going to lose the hydrogen and is going to leave you with 6 ADP plus and 6 phosphate. As that cycle continues, you're going to get 6 glyceraldehydes and 3 phosphate, and it continues at 3 carbons. And here you're going to lose a molecule of glyceraldehyde 3 phosphate. So instead of 6, now you have 5. You're going to lose 2 phosphates after this, and you're going to add energy again. Add 3 ATP, and you're going to leave 3 ADP is going to leave. And that's going to take you to 3 ribulose, which is it's going to be 1,5-biphosphate, and you're going to start the cycle all over again. So basically what you're doing here is you're gaining and losing phosphates, and it's basically a lot of transferring, all to keep the cycle alive, just so you can start it all over again and add the carbon.